Hello, good afternoon, everyone. Hope you had a good weekend, and I know we're all lacking an hour of sleep, but thank you for being on time and being here. We're gonna go ahead and get started. Hopefully, we can finish lecture 6 today, and we're gonna start on lecture 7. But looking at the schedule here, um, we're slightly behind. Um, I think we might have to move the quiz. I will let you know after today's lesson. We'll see where we end up being. We might have to move it to Wednesday or something, and actually have a class on Monday. We'll see. We'll see. I'll let you know. So let's get started. Previously, last week, we talked about various experiments that had led to, confirming DNA is the hereditary material. That was the Hershey and Chase experiment. There was Rosalind Franklin and Watson and Crick. They determined the structure of a DNA molecule being that double helix. And later on, there was also Messelson and Stahl, who had conducted experiment to show that DNA replication follows the semi-conservative model, right? And we also spent quite a bit of time talking about the enzymes and the proteins that are involved when it comes to DNA replication. So there is the gyrase, there is the helicase, and single strand binding protein and all those things. Or talk about the leading strand, lagging strand, Okazaki fragments, all these terms that I just mentioned. You should be familiar with them when it comes to the quiz time. And we ended by talking about the importance of having telomeres at the end of our DNA to delay the erosion of important genes located at the end of chromosomes. And we talk about cloning as well as telomerase, which is needed to reset our DNA length in the germ cells so that our next generation is going to have brand new telomeres so that they're not going to be born with shortened ones. And then we also talk about how turning on telomerase in a regular cell is a contributing factor of cancer. So today we're going to talk about the cell cycle and mitosis. We'll also examine what's going to happen if the regulation and the control in mitosis are not there, the consequences of something like that. So let's get going. Briefly, we learned about the cell theory back in lecture 3, and I'm not going to read all of these to you again. You already know it. So for the first half of this lecture, we basically talk about how DNA is the hereditary material that it's going to get passed on to the next generation when a cell divides. And the number 3 here says all cells come from the division of pre-existing cells. So when we need to have more cells, the only way we can get more is to use the existing ones that we have in the body and wait for them to divide. So what's the purpose of dividing cells? Well, we need to replace lost or damaged cells. If you have a cut, you damage some cells. You've lost some cells during the process, and you need to get new ones to replace it. So you need to do cell division. And in case I'm not being clear here, okay, so this is like the original cell. We call that the parent cell. Some people get confused. Okay, parent cell is not talking about the cells in your parents. It is cells in your body that is about to divide. And is going to do DNA replication, right? Like all the stuff we learned last week. That's a DNA replication, doubling the amount of genetic material. And then the cell is going to split into 2 daughter cells. Daughter cells. Okay, so this process of division is called mitosis. And that's what we're going to be focusing on for the first hour or so. So we need to do mitosis for replacing lost or damaged cells. We need to do mitosis for the purpose of growing and developing. When we were younger, we have much fewer cells than we do now. And we're smaller as well. So to physically get bigger, we need more cells. And those extra cells came from cell division. Mitosis or cell division can also be used as a way for reproduction. Now, humans and animals, we do not generally we do not use mitosis for reproduction. Definitely not in humans. Okay, animals generally don't do it either. Mitosis is reserved for asexual reproduction. Okay? So asexual reproduction is a type of reproduction that does not require sperm and egg fertilization. Right? The advantage is you don't need a partner for asexual reproduction. Only one individual is required. If you are the last individual left of the species, that's okay. You can still repopulate, okay, just by doing asexual reproduction. It is relatively fast and require less energy. The major disadvantage of this is when you are doing mitosis, you're essentially cloning yourself. We talked about cloning last time. So let's say we have the amoeba here, and it's doing asexual reproduction to make many copies of itself. All the amoeba that comes after will be genetically identical. The problem with a lack of genetic diversity is that if there is some kind of adverse changes in the environment and it's like a disadvantage for a particular genetic makeup, all of them are going to be affected equally. Let me give you an example, more concrete example. I'm sure by now you or someone you know have caught COVID, once or more than once, okay? And I'm sure you're also aware that some people have it harder than others, okay? So one of the reasons for that is because of our genetic diversity. Because everybody has a different genetic makeup, the virus caused some people to become more ill than others. If everything was the same, genetically identical, then the virus, if it's bad for one individual, is going to be bad for the entire population if they were all genetically identical, right? So we do not want things to lack genetic diversity because it's really bad for the population. If there's an environmental factor that's having a negative impact on one individual, the whole population can be wiped out. And that's actually what's happening with bananas, if you believe it. Okay, it turns out bananas that are grown in plantations are mostly grown by asexual reproduction. Plants are easy to grow asexually. Okay, you can break off parts of the plant, and then you can basically use that part to grow a genetically identical plants, right? And so a lot of the bananas in the banana plants in the plantations are genetically identical to each other. So there is a fungus that's circulating right now in places where we grew bananas, and it's killing a lot of banana trees. And because they're all genetically identical, the fungus has the same intensity of negative impact on all of them. So plantations are losing a lot of crops due to that. And if they don't do anything about it, the production of bananas might take a hit. And banana export makes up a lot of revenue for countries who very good climate and growing these kind of plants. If you want to know more about it, I'll post video to show you after class. Okay, it's on YouTube. It's quite interesting. It's like 10 minutes or something like that. Anyways, other organisms that can do asexual reproduction include starfish, or sea star. Some people call them sea star. If you break a starfish in two, you will have two starfish later on. It's going to be able to grow back. Okay, it's different from worms. Okay, some people you probably heard that worms, if the worm get cut, it's gonna grow back its tail. Okay, so it's true, but the tail is not gonna grow a head back, right? So that's not reproduction. That's just like regeneration. You gotta understand the difference. Plants and some other microorganisms, they are able to do asexual reproduction. Okay, let's do a quick recap with the structures of DNA. So on its own, when it's just like naked DNA, then it's just the nucleotides, right? The ACTGs connected together, and it's really, really long. Okay, all the DNA in the body, it's long enough to wrap around your body many times. Okay, like 6 or 7 times the size of your body, basically, if you're lining up all the DNA end to end. And there's no way to pack all of them in the nucleus if we don't wrap them around something. So we wrap them around special proteins called histones, and that is gonna form the chromatin. Okay, chromatin is basically DNA plus protein. Okay, IN. That's how you remember it. Now you can take the chromatin and you can condense it even further, and that's gonna give you the chromosome. Right? So chromatin and chromosome essentially are the same thing. It's just how compact it is. Right? The chromosomes are the most compact version of it, and you need to pack it into the chromosome form when you need to move them around. When do you need to move your genetic material around? When you do cell division. Okay, you need to make sure that you have equal amount of genetic materials, right, in your daughter cells. So you got to move the chromosomes around, and you need it to be very condensed. Think about moving to another apartment. You got to pack your stuff. You move and then you unpack them. So before you move the DNA, you got to pack the chromatin into chromosome, and then you can unpack it later. So there you have it. The difference between DNA, chromatin, and chromosome. Maybe have a list of these key terms next to you, because I'm going to introduce a few more later on as we go on, and it's only going to get more confusing. So how many chromosomes do we have in the body? I must have told you this before, but let's just ask this again. How many chromosomes do we have in our cells? Does anybody know?
23 pairs, 46.
That's right, 23 pairs or 46 chromosomes. So different organisms have different number of chromosomes, but humans, we have 23 pairs. Okay, 23 from mom and 23 from dad. Okay, so our mother would pass us 23 through the egg, and the dad passes 23 and through the sperm, and together, right, it's gonna make 46. Of the 46 chromosome, 22 pairs of them are called autosomal chromosomes, or autosomes for short. So these autosomes, the 44 autosomal chromosomes, they have instructions on them, genes on them, that determines everything about us except for the sex. Okay? So, your eye color, your blood type, to a certain extent, your height, okay? What kind of ears do you have? Is it attached or is it detached? Right? All these things are all encoded within the autosomal chromosomes. Okay? 44 of them. Number 45 and 46, those are the sex chromosome. In male, it is XY. In female, it is XX. So did you know by default, the embryo, when it's growing in the mother, right, the fetus, by default is gonna be female. But if it's genetically XY, then the Y chromosome gets turned on during embryonic development, and that causes the embryo to start to develop into a male. Okay, if the Y doesn't get turned on, then it will continue to develop as a female, and that sometimes can happen. There's a
Vincent, how
question.
does it get turned on?
How does it get turned on?
Yeah.
Yeah, there's a gene called SRY. We will maybe talk about it later, but
Oh,
yeah,
okay.
yeah, it's a good question. There's another message, private message. Why do humans have 46 and other organisms have 48? Yeah, there's like I have a slide coming up that shows you some organisms have more, others have less. We don't know, okay? It's not dependent on the size either. Like goldfish is pretty small, but it has like 100 chromosomes in it. Okay, so there's no correlation between the amount of genetic material an organism has versus how many chromosomes it has. It doesn't even have to do with like complexity or intelligence. Okay, something like a plant can have as many as like thousands of chromosome in some cases. So we don't really know. Yeah.
Yes, so Vincent, I have questions. So, what about gene? Is it kind of a certain sequence of DNA
Yeah,
proteins or
so
something like
yeah, we're gonna talk more about genes later, but this is the chromosome, okay? The chromosome number 2, for example. So it's DNA, right? Wrapped around proteins and whatnot. So from one end is like A, C, T, G, dot, 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 all the way to the other end. It's like it's just made up of 4 letters, right? A, C's, T's, and G's. Now on the chromosome, like a section, right? That's what we call like a gene. A gene is a segment of the chromosome that has the instruction to make a protein, basically.
Okay, thank you.
Okay, yeah. So we're gonna talk more about that later on in lecture 7, but that's like the short version. Think of the chromosome as a chapter in the book, and a gene would be like a section, like section 2.4 in the textbook. Okay, that's the relationship. So 22 pairs of autosomes, 1 pair of sex chromosome. So is it the father or the mother that determines the sex of the baby?
The father.
That's right. The father has the X and the Y. Half of the sperm will carry the X, the other half will carry the Y. And if the one carrying the X fertilizes the egg, it's going to be a girl. If the sperm carrying the Y fertilizes the egg, it's going to be a boy. My grandmother, she's very old-fashioned. She's like 90 something now, okay? But sometimes she would every now and then she would sit next to me and start asking me, how come your wife doesn't give you any son? Because I have 2 daughters, right? And then I would try to explain to her, it's not her fault, it's actually my fault, right? It's the guy that is responsible for determining the gender. She just wouldn't listen. She's like, no, that's not true. I know, because I have 3 sons. It's always the wife. Well, despite of what most people traditionally thinks, it is the male that's responsible for the gender of the child. Okay, so moving on, the cell cycle. Right? The cell cycle is basically the life of a cell, if you would. Okay, it includes the whole time that the cell is preparing, it's growing, it's performing its function, and then ultimately it's going to divide and create 2 daughter cells. So right up to the point of division, we call that interphase. The cell is just performing its normal function. Right? If it's a muscle cell, it's going to be contracting. If it's liver cell, maybe it's going to be detoxifying your blood, right? It's growing, it's preparing for mitosis. And then the mitosis is the actual cell division where it splits into 2 daughter cells. Now, here in purple, this whole portion, okay. Oh, it says right here. Sorry, I didn't notice. That's the interphase. Okay, it's roughly accounting for 90% of the cell's life. Okay, now that's like a generalization. Right? You gotta understand, not all cells divide in the body, and when they do divide, they don't all divide at the same pace. Some cells, like the skin cells, well, the surface of the skin is dead, right? But like to get new skin that comes from the below, they divide pretty quickly, okay? Every couple weeks you get new ones, okay? And some organs like the liver. Did you know if you donate like your liver, like if you donate like a third of your liver, it's gonna come back. Like you're gonna be able to regrow your liver, right? So those cells have very high proliferation rate. They can divide rapidly. Okay. Can anybody give me examples of cells that don't divide? Like if you damage these cells, they don't usually come back. They don't come back.
Brain cells?
Brain cells, that's right. So, you know, the brain cells, the neurons, if you damage the neurons, they don't come back, right? And another part of the body that don't really divide when you damage the cells, anybody?
What about the eyes? Do we have cells in our eyes?
Yeah, we do have cells in the eyes, but they could divide a little bit. Yeah.
Okay.
Yeah. The heart, okay, heart cells. That's why people who have heart attack, their heart don't fully recover, right? It doesn't heal itself because of like they don't divide. That's why there's no heart cancer. If the cells don't divide, they cannot give rise to cancer, right? Cancer, as I told you, is uncontrolled cell division, right? So there's no such thing as heart cancer. Brain cancer,
Muscle
though.
cells.
Yeah, muscle cells can divide. Yeah, every time you work out, you actually tear the muscles, and the way that you get more muscle is by creating new muscle cells. So those actually divide. What was I saying? Yes, brain cells. There are actually two types of brain cells. The neurons, they don't divide, so those don't give you cancer. But then there are other cells that supports the neuron that are also in the brain. Those are the ones that can divide and give cancer. Anyways, typically speaking, a cell spends roughly 90% of its life in the interphase, and the actual division is about 10%. And as you can see in this picture, the interface is further divided into different parts. Okay, there is the G1 and G2. Like getting a driver license, okay? G1, G2, and then there's an S phase in between. And the mitosis also is separated into various stages. So let's take a closer look at the different parts of the interface. There is the G1, S, and G2. And along the way, there are various checkpoints to check to see if the cell is ready for cell division. Hold on, let me just check my notes here because I reorganized some of these notes recently. I just want to make sure that I am telling you the right stuff at the right place. Okay, I am. All right, so here, G1. Okay, what is G1? G1 is just growing and preparing. Right, the cell is getting bigger and bigger. If it's too small, there's not enough cytoplasm to divide, right? And then it's preparing for DNA synthesis. Okay? Before you divide, you have to copy all your DNA, right? So it's growing, it's growing, it's growing. At the end of G1, though, we have a first checkpoint here. Checkpoint. You need to write this down, okay? Checkpoint number 1. Okay? And what are you checking for? Well, you are checking to see if the DNA has good quality to be copied. What do I mean by that? So a couple of examples. It's actually kind of written here, but I want to write it again. So DNA error. Right? Is there any mistake? Are everything base paired properly? Like are the T's with the A's, are the C's with the G's? Right? Is there any mistake, any problem, any breaks in the DNA? It's possible to have breaks in the DNA sometimes, right? So any DNA error, any DNA damage, right? And you might also want to check your telomere length, right? Are they long enough to go for another round of copying. Because remember, every time you copy your DNA, it gets shorter, right? So like, are they long enough to sustain another round of division? So you will check all these things. Right? If there are errors, you will try to repair it. Sometimes you can repair it, sometimes you cannot. Same thing with the damage, right? So if everything is okay, or if you're able to repair it, then it gives them the green light. And go ahead, copy your DNA. And that's what the S phase is for. S is for synthesis, okay? You do the DNA replication. Right? So all 46 chromosomes get copied. And then afterwards, we have another checkpoint. Checkpoint number 2. Checkpoint number 2, right there. Right. So again, we're checking for the same thing, right? Any DNA error, damage, right? Did you copy everything? Okay. So you have to check before and after. Right? Before and after you copy, you gotta check the quality and everything. And if everything is okay, then it's fine. You will be able to proceed to mitosis. Okay, you'll prepare a little bit more, and then you will proceed to mitosis. If the errors cannot be fixed, if there is some kind of irreparable mistake or damage, then the cell will not be permitted to divide, and they might do programmed cell death, right? Which is apoptosis. We learned about this way back, right? With the lysosome and stuff like that. It's also possible for the cells to enter senescence, right? To become senescent means to not divide anymore. Maybe the cell is perfectly fine. There's like, no damage or anything. It's still functioning, but it has divided too many times already. Telomeres are too short. So, it will enter growth arrest. It's not going to be permitted to divide anymore. Any questions about the checkpoints and what they are for, or of anything at all? Okay. So here it is again, the interface, and it's going through how the cell divides and everything. I think it's probably better if we should we draw this out? I'm thinking. Let me see. Yeah, maybe let's do that. Let's draw it out. It's probably a little bit better for your understanding, even though it takes a longer time. Right. So here we go. This is just interface. Okay. So here is the cell, and in the nucleus, we're gonna have we're gonna have some DNA. Okay, but at this point, it is still in the form of chromatin, so you can't really see them. Okay, so these are still chromatin. Okay, and remember, an interface is not part of the mitosis. Okay, this is just getting ready, right, with the G1 and the S and the G2. Anybody remember what these things are? It's from lecture 3. Anybody remember what those are?
Centrioles.
That's right. They are centrioles. Yes, very good. Okay, centrioles, right? So what happens is, you can draw it, and then you can have your PowerPoint next to you. I don't want to rewrite the PowerPoint stuff. But basically, at this point, you would be doubling your genetic material. Okay. And so we're going to talk more about how this happens later on. But basically, if you have, let's say, chromosome 1 here, chromosome 1, after duplication, you're gonna have another one here. Okay, hold on. Let's try that one more time. Okay, so you have two chromosome 1, right? Chromosome 1 and chromosome 1. This is chromosome 1 from dad, like your dad, and this is chromosome 1 from your mom, right? And in the cell, when you try to divide, you're gonna have to do DNA replication, and then you end up having like this. Okay, each chromosome will now have an extra copy. And this is where things get a little bit complicated. Okay? You see two chromosomes here, right? Two chromosome 1. How many chromosomes do we have here? What do you think?
2.
Okay, yeah, that's the correct answer, okay? But many people would say 4 because they see 4 here, but actually it's still 2, okay? If they are stuck together, then we count them as 1. So this is also 2 chromosomes, even though you have duplicated. Do you understand that? Okay. So each of the chromosome has made a copy of itself, okay? And they're stuck together. Right? So this is still 2, even though now you have like 4 pieces, right? Okay. So some extra terms for you. The center, where the 2 chromosomes are stuck together, that is called the centromere. It's the location where chromosomes, where we would say duplicated chromosomes are connected. Okay. So this copying happens nearing the end of the interface, right? During the S phase of the interface specifically. All right, so I just wanted to show you that quickly. Let's go back to the PowerPoint here. Right. In this picture, this is the real picture of what's happening in a cell. Okay, over here. You can see that's the nucleus. All these yellow strands, they are the cytoskeleton, which in this case is the spindle fibers. And then the red is just where the cytoplasm is, okay? All right, so I'm gonna read some of these things to you. Again, interface is like 90% of the time, right? It's performing its regular function, and nearing the end, the cell prepares to undergo mitosis, right? Specifically, DNA will duplicate itself. So right now, you still see this is mostly chromatin, right? If you take a look at the real picture, I don't see anything. Like in the nucleus, it's just a sea of blue. I do not see the individual chromosome. So later on, then we go on to the mitosis, which is separated into 4 phases. Okay, so prophase, metaphase, anaphase, and telophase. The order is PMET, right? PMAT. So let's take a look at what happens in each of the phases. So prophase, right? Early on in prophase. Remember, at this point, the chromosomes have already been duplicated. Okay, and you can start to see the chromosomes. You can start to see like they're almost like little noodles, right? So the chromatin has started to condense and become the chromosome. DNA replication is completed already. And it says here the sister chromatids are held together at centromere. So going back to our picture here, the chromosomes that are stuck together, they are exact copy of each other, right? So we call them sister chromatids. Okay, sister chromatids. Let me just clarify this a little bit more. It's maybe a bit confusing if I use the same color. I'll explain all these like later on again, okay? But let me try that one more time. Give me a second. You know what? It's probably easier if I just redraw it. Okay. So you have to understand, you know, even though we have 2 chromosomes for each number, like 2 chromosome 1, 2 chromosome 2, and so on and so forth, the chromosome 1 from mom and chromosome 1 from dad are actually not identical, okay? Because, for example, like one parent might have type A blood, one parent might have type B blood, then those information are going to be different on the chromosomes, right? So the blue one here represent the one from dad, and the red one represent the one from mom. After you duplicate the DNA, right, the ones that are identical are going to be the sister chromatids. So like, you know, the red one here and the red one here, if they're stuck together, that's the sister chromatids. Okay, they are identical in every way. Whereas these 2 chromosomes, even though they're both chromosome 1, there are subtle differences between them, depending on who your parents are, right? And maybe they both have brown eyes, then the eye gene is going to be the same. But, you know, maybe one of them has dimples and the other does not have dimple, then it's going to be different. Okay, so there are variations. Okay, so there are a lot of words here. Okay. Do you guys understand the difference between chromatin and chromatids? Okay, some people pronounce this like chromatin to distinguish it from chromatids. Okay, but this is basically, okay, so yeah, one more time. This is basically DNA plus protein. Okay, when you wrap them around the proteins, you get the chromatin. You put them into the nucleus. The chromatids, TID, that's what you get when you copy the DNA. So here's the chromosome 1. When you copy it, you have 2 copies that are stuck together. They're exactly the same. So like this one and this one, we call it chromatids. Okay. It's like, you know, if I have a pencil and then I make a copy of the pencil, so now I have 2. These are exactly the same and they're stuck together. Chromatids, chromatids. Okay.
Okay, Vincent. So to understand you better, so in the nucleus now, we have proteins.
We always had, yes.
Yeah. Okay. Yeah. So now, so the DNA, do they always, you know, like in their natural form, like around the chromatin, or?
Okay, so DNA on its own is just a double helix, right?
Yeah, the double, yeah.
That doesn't exist in the cell. Okay. They always wrap, so like, you know, if I continue drawing them, they always wrap themselves around these proteins, right? So they wrap around it and then wrap around it and then wrap around it, right? So this form that I'm drawing here, it's like the spool, the thread around the spool, right? Like the sewing thread that I told you about. Okay. So this is going to be chromatin, okay? Chromatin. Okay. And then after you copy them and you need to move them around, they condense more. So they coil up around even more more proteins. Okay. And then this is called a chromosome. Okay, but they don't coil up all the way to chromosome until after you copy them. That's why when you see chromosome, they always look like an X. You know what I mean? Right? They always look at X. So that's a chromosome. And where they stuck together, that's the centromere. Okay? Not to be confused with the, not to be confused with the things at the end, right? The things at the end, that's the telomeres, right? Okay. And this copy and this copy, they're the same. So we call them sister chromatids. Okay?
Professor, excuse me.
Mm-hmm.
Professor, you say the chromatin, there is DNA wrapped around the protein, but
Proteins, yeah.
chromosome also there is protein inside
That's
chromosome?
right. Even more protein because you gotta pack it even more, right?
Okay.
Yeah.
Okay. Thank you.
So chromatin
sorry.
and chromosome are both DNA with proteins, but this one is like even more packed. Okay, you crunch it down even more. Sorry, yeah, go ahead.
So
Aminette?
so we said these centromeres, is there any chemical bound here, or just it's attraction between two?
Yeah, good question. Yes, there are a bunch of proteins there that helps them stay together. Yeah. So they don't just get attracted to each other. There has to be like protein glue that binds them together. Okay.
Okay, thank you.
Yeah. Aminette, do you have a question? I saw you on mute. Or was
Oh,
that...
no.
No, you good now? Okay. All right. Now, I know I'm sorry, not doing a very good job explaining this, I feel like. I'm just gonna go back here. You don't have to go back. Okay, just watch. Look here. What are you looking at? Tell me, what is this? Is that DNA? Is that chromatin? Is that chromosome? What are we looking at, people?
Chromosome.
That's right, because it's two of them already, right? They are only gonna get that condensed after you divide. Right? After you, sorry, not after you divide, after you copy the DNA, right? So each of these, that and that, they are the same. They are identical. We call them sister chromatid, right? TID, sister chromatid. And then the whole thing is a chromosome. And I know this is a super small picture, but like right there where they're attached, that's the centromere, right? Centromere. Is this a little bit better? Any follow-up questions?
So Vincent, we don't see this. Sorry, I interrupt somebody, I think.
No, go ahead. Yeah, go ahead.
So in the nucleus, we don't see like this. When it's gonna divide, we see this chromosome like this, or no? I'm
Sorry,
confused.
one more time. What's the question?
So in nucleus,
In the nucleus, yeah.
we don't see like these chromosomes, right? We
That's right.
see
So yeah,
chromatin.
that's exactly. So look back here, right? Like at the in the interface, look at the nucleus. Do you see any chromosome or anything like that? No, right? Like it's just like very, like I don't see anything, literally. Maybe some lines there. Why do we not see it? Because they are still in the chromatin form, right? The TIN, chromatin form. Okay, we don't see them. It's very fine line. You don't see them. And then they will duplicate and copy themselves, which is fine. But then when you're ready to move them in the cell, then they start to condense, right? Then the chromatin coils in the visible chromosome. It's easier to see them when they coil up, right? Do you start to see them now over here? Like if you look at the screen, you can see the outline of them, right? I hope. Maybe? No? Yeah?
Yeah.
Yeah. If I go on, now it's very clear, right? Okay. So it's starting to get more clear, and then it's very clear now. So again, this is chromatin. Most of the cell, when it's not dividing, it's gonna exist in this form. It's only when you start to divide, after you have copied the DNA, then it becomes a chromosome. Okay? And then it's the nucleus envelope is gonna dissolve, and then all these chromosomes are gonna come out. Okay? Then the spindle fibers so a couple of things here, right? The nuclear envelope starts to disappear. Of course, it has to disappear. Otherwise, how can the DNA come out, right? And then the centrioles, they produce the spindle fibers, and you can see the centriole actually move towards the opposite side of the cell. And this is still prophase, later on in the prophase. The nuclear envelope will be completely dissolved. You don't see them anymore. And then you will have a lot of spindle fibers that are coming out, and some of them will attach to the centromere. Right, you see that one attached to the centromere, attached to the centromere, like so. That's what happens up to prophase. I'm sorry if I'm making this very confusing. I will post a video for you to watch this as an animation. Maybe that will help a little bit. But any other questions at this point before I make it worse? Okay, so after prophase, remember PMAT, right? So P, you can think of P for preparing. Okay, P for preparing. And now we're gonna have metaphase. So in metaphase, the spindle fibers help line up the sister chromatids at the center. Okay, so you see before one slide earlier, they are kind of everywhere, right? But look, the centromere is gonna have a spindle fiber attached on both sides, and there is this like tug of war that goes back and forth for all the chromosomes until they all line up in the middle. Okay, so M is for middle. Everything lines up in the middle. So that's one chromosome, two chromosomes, so on and so forth. So in reality, there would be a total of 46 chromosomes, all lined up in the center. Okay, so you can see here, right? That's the cell. That's the centriole, and then all the chromosomes in the middle. And the green lines, those are the spindle fibers. After you line them up in the middle, then you're going to split them apart. Right? So A for apart. You're going to pull them apart. So remember that and that, they are sister chromatids, right? Sister chromatids. That is now separated. Once you separate them, they call them daughter chromosomes. But all that means is that they are actually identical copies. Okay, so the centromere is no more. It gets separated, and we have 4 chromosomes here, 46 really, but they only draw 4. And then after you split it, right, each will get half. But because they're separated, we count them separately now. Right, so it's like 8 chromosomes here. And then you pull them to the opposite end. And the last part is telophase and cytokinesis. This is basically the reverse of the prophase. So now we finish moving the chromosomes. They will start to uncoil and become chromatin again. The nuclear envelope will start to form around the chromosomes. We do not need the spindle fibers anymore, so they start to disappear. And then the cell will divide and split, basically split the cytoplasm half and half. And that process of splitting the cytoplasm is called cytokinesis. And that's all. That completes the mitosis. So let's take a look at this. This is onion cell. And we apply a little bit of stain here to highlight the nucleus. Based on what you just learned, can you tell what phase this might be? Is it prophase, metaphase, anaphase, or telophase? Just take a guess. What do you think this is?
I think metaphase.
Yeah, and how do you know? What gives it away?
the chromosomes are lined up.
In the middle, right? That's it. Right. So M for metaphase is all in the middle. Right. This is the cell. Plant cells are very structured, right? They're more rectangular in shape than like circle for animals. Okay, what about this one? What about B? Okay. What do you think? You can type the answer if you don't want to.
Anaphase.
Anaphase. Okay. Anyone else? Do you guys think this is anaphase, or do you agree? Do you think it's something
Yeah,
else?
I think it's anaphase. Yeah.
Yeah. Anaphase. Yeah, this is anaphase. Very good. Right? Because the chromosomes are being pulled apart, right? You see, it's separated. It's being pulled to the opposite side. Anaphase. Right? Here is another anaphase. You see, another anaphase. Do you see another anaphase somewhere? Maybe.
Yeah, down the last.
Yeah,
Yeah.
that's right. There we go. Very good. Okay, anaphase, anaphase, anaphase. Okay. All right. What about this one? This one is a little bit harder to see, but if you really look closely, you can see there is starting to have a little line in the middle. And the thing is, like, I don't see the chromosome anymore. They're more like concentrated, right, on either side. So this is actually telophase. Right? Telophase. Starting to become two cells. Okay. This one. What about that one?
Early prophase.
Yeah, early prophase. We would just say prophase. That's right. Because I see the chromosome, right? But they're not really out of the nucleus yet. Okay. Something like that would be interphase. Right? Interphase. In fact, this whole row is like mostly interphase, right? Okay. Is that better? Well, I'm not going to ask you to describe the process. If you're able to identify them and remember some of the key things, you should be okay. Questions before I move on? Okay. During which phase of mitosis does the nuclear envelope dissolves? Let me do the poll for you. Okay, that is the prophase. Okay, prophase. Prophase. Let's try the next one. Okay. Go ahead.
Vincent, there is no pole for this one.
There's no what for that one?
Pole, like the pole.
Prophase?
No,
Sorry.
the pole.
Oh, I didn't launch the poll. Oh, I'm so sorry. Yeah, I got this. I got an email. I was checking. Sorry. It's okay. I'll do this one with you guys. Okay. So a cell has 8 chromosomes at metaphase of mitosis. How many chromosomes will it have during anaphase? Okay, so 8, right? So 1, 2, 3, 4, 5, 6, 7, 8, right? All lined up in the middle, huh? And then anaphase means you're gonna separate it, right? So once you separate, you count them individually, right? Which is how many now?
16.
16, that's right. That's the right answer. Okay? Very good. Very good. You can read this later, okay? No short answer on this quiz. Yeah, don't worry. But you can read this and think about it. Okay, we gotta move on. Sorry. Okay, next thing is confusing. Okay, next thing is very confusing. But fortunately, I am better at explaining this. You know, it was difficult for me to explain the mitosis online because like it's better to see it as a video, as an animation, right? So that's why that was a little bit clunky, and I do apologize. Okay, now this next thing is confusing, but it doesn't require as much visualization, right? It's just like factual stuff. So even though it's confusing, I should be able to explain it a little bit better. All right, but you gotta pay attention. All right. Oh, thank you for the encouragements. Some of you saying, you know, don't worry, I'm doing okay. Thank you. Okay, so the cell cycle and the checkpoint. Now, I was telling you about the cell cycle and the checkpoint being important to check for the quality of the DNA both before and after you copy the DNA, right? And so these checkpoints are actually done by proteins. And as you know, you should know by now, proteins, the instruction to make the proteins are encoded within the DNA, right? Encoded within the DNA. So, which means if you have a mutation in the DNA, and we'll learn way more about mutation later on, but for now, mutation is a permanent change in your DNA, and that could happen just by chance or by, you know, there are various things that could cause mutation, right? Like UV light, that's why you have to put it on your sunscreen, like smoking. All these things can cause permanent change in the DNA. So if you change your DNA, then the instruction to make the protein is going to be wrong, and that will change your protein, right? And if that protein that was affected happens to be one of these checkpoint proteins, then we are going to have a problem. Okay? So there are two types of checkpoint proteins. We call them regulators. Two types of regulators. We have the ones that are called growth factors. And as the name suggests, growth factors are gonna promote cell division. Think of them as green lights. Okay, green lights. Right. And we have growth inhibitors, which again, as the name implies, they inhibit mitosis. Okay, so those are the red lights, okay? Now, the reason why I draw two green circle and two red circle is we have two sets of chromosome, right? So if we have two chromosome 4, for example, and I have a gene on chromosome 4 that acts as a growth factor, I would have two copies of it. Each chromosome 4 is going to have a growth factor, right? So one green light on each. And then when they light up, it tells the cell to divide. Same thing for the growth inhibitor. Right? I would have two red lights to stop the cell from dividing. Okay? So in the body, there is not just one growth factor or one growth inhibitor. You have hundreds of them, and they all work together. Okay? Now, in general, in a general sense, you have to have multiple mistakes before a cell turns cancerous. Okay, but I'm just gonna keep it very simple. Okay, I'm gonna say, you know, with one mutation, we're gonna see what the worst outcome is going to be. So allow me to explain. There is something called a proto-oncogene. Proto-oncogene. A proto-oncogene is a gene that normally helps the cell to grow. This is a regular gene. But okay, let's take a step back. Does everybody understand what a gene is? Okay, what is a gene? I briefly explained it to you earlier, but what is a gene? A gene is, yeah, sorry, go ahead, please.
Okay, a part of the chromosome.
And what does it do? Instruction to do what?
To produce more protein.
That's right. Instruction to make protein. It's the recipe to make the protein. Okay? So a proto-oncogene is the recipe to make a protein that will help the cell grow. In other words, it's the recipe to make the green light. Okay? Now, when the proto-oncogene, the normal one, when it mutates, when they have a permanent change, okay, this is permanently changed. And your DNA can permanently change again, just because of bad luck by chance, or because of external factors, you know, like alcohol, smoking, exposure to carcinogens, okay? When it changes, then the proto-oncogenes become a bad gene, okay? When it becomes bad, it is permanently turned on. Permanently turned on or permanently activated. In other words, it is going to be a green light that's always on. Okay? Think about traffic. If it's always green light, what happens? The cars are just going to keep going, right? If it's always green light, then the cell division is going to keep on dividing. They don't care. Okay. If there is a mistake, who cares? Copy the DNA anyways and hope for the best. It's going to divide. And so this mutated version of the gene is now called an oncogene. Oncogene. Okay, so proto-oncogene is the normal. Proto means like before. And oncogene is the bad one. What do we call cancer doctors? Let's also start with an O. What do we call them? You have to see a
oncologist.
That's right, an oncologist. Same word, oncogene. Right? So that's a permanent green light, and it's gonna tell the cell to divide. And this out of control cell division is what leads to cancer. Are we okay with this before I tell you the second one? Okay.
Vincent, the mutation is like changing the protein in the strand.
Yeah, not changing the proteins, but changing the DNA. Mutation is a permanent change in the DNA. Okay. But then don't forget, in DNA. Okay. But then don't forget the DNA is the instruction to make the protein, right? So if you change that, then your protein is going to be different. Right? It's like the recipe to make a cake. If you cross out the sugar and then substitute it with salt, your cake is going to turn out completely disastrous, right? So same thing. If we change the ACTG in the DNA, the protein that we make in the end, it's going to be something different. Okay? And in this case, it's going to make the green light always on. And that's going to be problematic, right? Now, the other regulator is the tumor suppressor gene. These tumor suppressor genes, as the name suggests, they suppress tumor formation by slowing down cell division, right? If there are DNA mistakes, they're going to repair it, okay? And if the cell needs to die, it's going to tell it to die and not divide. So these are essentially genes that makes the red lights. Okay? They're instruction to make the red lights. When tumor suppressor genes don't work properly, cells can grow out of control. Okay, what does it mean by don't work? Right? That means you have a red light that does not turn on. Okay? So if you don't have a way to slow down the cell division, if you don't have a way to tell them to stop because the red lights are not turning on, then they are just going to keep dividing and dividing, right? So there are two ways that could lead to uncontrolled cell division. A oncogene, which is a green light that's always on, or a tumor suppressor gene that's not working anymore, a red light that does not turn on. Okay, so a combination of both of these things can really send things off the cliff, and they could really start to divide rapidly which contributes to a tumor. Now, typically, the proto-oncogene is what we call a gain of function. Okay? Gain of function, to put it simply, is always on. The green light is always on, as in. Okay? In this case, only one mutation is enough to cause the cells to become cancerous. Okay? Because look, I told you we have two green lights, right? Two green lights. Okay? And I cannot do this in an animated way. Just look at my hands. Okay, two green lights. This is called on, this is called off, right? So normally, right, when you want to tell the cell to divide, you turn them on and the cells see the green light both on, and then they will divide. And then when you don't want them to divide, you turn them off, right? Green light is not on, don't divide. But if you have one that is permanently on, even the other one is working properly, on and off as it should, it doesn't matter because this one is always on, is always green. The cells will see the green signal and be like, okay, go, 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 go. They will pay no attention to the other one. So just a single mutation on one of the copies, even though you have two copies, is going to be enough to send the cell into this uncontrolled cell division, man. Yeah, it's as if like driving and then you have a stuck accelerator. Yeah, the gas pedal that you step on to tell the car to go. If that thing is stuck, that's it. There is no way of stopping it. Okay, like it's just gonna crash. Okay, what about tumor suppressor gene? Okay, tumor suppressor gene has what we call a loss of function. Okay, it does not turn on, or we can think of it as like always off. Okay, so again, think of the red lights, right? Think of red lights. Okay, two red lights because you have two chromosomes, right? Two genes for every protein. So we have two red lights. Okay, if you want the cell to stop, what do? You turn on the red lights, and then they see the red light's on, don't divide. Okay, but let's say one of them is busted. Okay, and it doesn't turn on anymore. It's always off. Right. So this one is still okay. This one turn on. Okay, don't divide, and then you can divide now. Don't divide. It's only when this one also permanently gets shut off, then there's no more red light. There's no backup, right? And then the cell will divide. Okay, it's kind of like there are two ways to brake. You use your regular brake, the one that you step on, or you can use the hand brake, right? I've never been in this situation, but like, you know, conceptually speaking, if your foot brake is not working, if I want to stop the car, I'm gonna try the hand brake, right? Okay, so there's two ways of stopping it. So both of those have to be not working in order for it to be out of control. So again, let's take a step back. Do you understand the gain of function, loss of function, tumor suppressor, and proto-oncogene? Any questions at all? All right, so here is another point that's important. People born with only one functional copy are more likely to get cancer. One functional copy of tumor suppressor. That's what it means. Okay. So look, we have many cells in the body, right? Many cells, and each cell would have two copies of the tumor suppressor gene. Okay? It will have two copies of every gene, anyways. But I'm focusing on tumor suppressor gene, right? So let's say by chance, one of the tumor suppressor gene in one of these cells get mutated. Okay, and that one doesn't work. Now, is that going to have uncontrolled cell division? Answer me, yes or no? What do you think? I have one hit now for the tumor suppressor gene in one of the cells not working. Is that enough to create uncontrolled cell division? What was it? Sorry, I saw someone unmute briefly. I didn't catch who it was. Do you guys understand my question?
Yes, sir.
Yeah. So is this enough to create uncontrolled cell division? No. No, right? Okay. Because you still have one functioning copy, right? And you still have one functioning copy of the red light. So what's the chance of having the same mutation in the same cell? Well, the chance is very low. Remember, we have trillions of cells. Like if I have the mutation in this cell, that's still okay because this cell also has like a backup copy, right? It's only when you have 2 hits in the same cell, then it's gonna become uncontrolled and grows into the tumor, right? So that happening on its own is really unlikely. Okay, it's sort of like closing your eyes and then randomly throwing an egg to the wall twice and hitting the same spot. Chances are low, right? But some people are born with, let me finish this and I'll answer your question. Some people are born with only 1 functional copy, which means at birth, all the cells already have 1 X in them. So now for this person, all right, another change in any of the cells, like this one, is enough to turn it into a tumor, right? Because only 1 brick is working to begin with. So as soon as you lose another copy, then that's it. You don't have a backup anymore. What was the question? Sorry.
So, mutation in both genes in one cell is enough. I mean, if one cell gets out of control, it is enough to make a tumor or cancer.
That's right. So like, look, this cell has both copies of the tumor suppressor gene mutated, which means there is no more red light in this particular cell. And this one is basically going to divide, and it's going to take over, right? Right. All the other ones are normal, but this one is going to take over. Do you understand that?
Yes, yeah. Okay.
Yeah. But this is only for tumor suppressor, right? For oncogene, a single mutation is enough, right? Because if one of the green light is always on, then the cell is going to go, right? Okay. So here, mutation and cancer. Cancer, some of them are inherited, meaning it gets passed down from the previous generation. There are certain mutations that makes a person more likely to have certain kinds of cancer. And then there are some that are acquired. Okay, there are environmental exposure that could increase your rate of getting cancer. And there are also certain activities that a person can do that might increase the risk of getting cancer. But now, even though I was telling you, right, like, 2 hits and you're gonna get a tumor or cancer, or like this sounds even more scary, 1 hit and you're gonna get cancer. But as I was saying in the beginning, right, I'm oversimplifying things. There are many backups and redundancy in the system to prevent this from happening. In reality, you actually need to have more than 1 mutation, generally speaking. Okay, and you have to have multiple red lights that are busted and multiple green lights that are permanently on. Then it's gonna cause the problem, right? Now, what I was saying is when someone has an inherited form of the gene that's not working, then the cells already start out with 1 bad copies. So it makes it easier and earlier for the person to accumulate enough mutation to cause cancer. That's why cancers that are inherited, that are passed on from one generation to another, tends to happen earlier in life than cancer of the same type that are not inherited. Okay? Even cancer of the same type have different categories, like breast cancer. Some are inherited, some are acquired. Those that are inherited, that tends to happen earlier in life than the ones that are not inherited, right? For the reason I explained to you, right? There is also the concept of penetrance. Okay, penetrance is the idea that talks about how likely will the gene be manifested? How likely will the gene be expressed when you have it? Okay, believe it or not all your genes are going to, turn out to be problematic when they mutate. Okay, sometimes people can have a certain gene for a particular disease, but they don't actually end up having the disease, right? So that's what we call incomplete penetrance. If a disease has like 100% penetrance, it means if you have the gene, for certain you're gonna have the disease. Okay, 50% penetrance means there's a half chance that you're not gonna have the disease, even though you have the mutated genes. Okay, so in general, inherited mutation leading to cancer have incomplete penetrance, meaning not everyone with the mutation will get cancer. Okay, so just because something runs in the family and you happen to inherit the bad gene does not necessarily mean you're gonna have it. There's a higher chance you might have it, but it's not like guarantee necessarily, right? So people like to do those genetic tests on themselves, right? With I don't know what the companies are, like 23andMe or something like that. People like track their ancestries and test their DNA. So I personally, I don't think that's something that I would do, because like what if it comes back and say I'm at risk of getting this and that, right? It could be sad knowing that, but at the same time, right, those are not definitive, right? Just because you inherit the bad genes, right? The concept of penetrance means you don't necessarily gonna have it. Now I want to give you an example of one of these genes, okay? It's called the BRCA1 gene, which stands for breast cancer. That's where the BRCA comes in. 1 gene. It's the first one they discover associated with the breast cancer. Now this gene is responsible for repairing DNA damage or destroying cells if the DNA cannot be repaired. So due to the nature of its function, this would be considered as a tumor suppressor gene, right? Because it's stopping the cell from dividing, right? Stopping the cell from dividing. All right, so BRCA1 gene is a tumor suppressor gene. About 12% of women in the general population, statistically speaking, will develop breast cancer sometime during their lives. Right? 12% of the population. These are the non-inherited version, right? Just by chance. However, women who inherited a harmful copy of BRCA1 gene, the tumor suppressor I was just telling you about, if they already are born with one mutated copy, then the chance of them developing breast cancer by the age of 70 is as high as 55 to 65%, depending on the ethnicity, for example. Right? The range might vary. But if you are born with one mutated copy of BRCA1 gene, you can see the risk is substantially higher than the general public, right? And I already explained to you why, right? Because now, if the person only has one functioning copy and that one also goes bad, then you're gonna get the cancer. But if you have 2 good copies, it takes 2 hits in the same cell for this to become problematic.
Vincent,
Yeah.
if the BRCA1 gene is a tumor suppressor gene, then why is it possible that they have it?
Why is it possible they have it?
That this 12% of women will have it.
Okay,
we develop breast cancer with this one.
Yes. So maybe I wasn't being clear. Everybody has the BRCA1 gene. Is that okay? Yeah.
Okay.
When it's functioning normally, it's a tumor suppressor gene, right? It stops the cell from dividing. Okay. So everybody has that. The problem here is for regular individuals, they have 2 copies that are working. So it's only when both copies are busted, when both copies are not working anymore, then the person would get the breast cancer, right?
Oh, okay. Thank you.
Yeah, but if you're born with one harmful copy already, then you only have one chance. You don't have like a backup, right? Does that make sense?
Yeah. Yes.
Okay, great. So you guys might know her, right? She is an actress, Angelina, famous actress. Her mother died from breast cancer. Okay, and she did genetic testing and found out that she has the BRCA1 gene. Right? So as a preventive measure, okay, she underwent double mastectomy. Okay, so basically removal of the breast, right? Because she doesn't want to have the cancer, right? As a preventive treatment. Right? Again, it doesn't mean she's gonna have it just because she has the BRCA1 gene, but you know, seeing what her mother went through, you know, she wanted to take control and preventive measures so that she doesn't have to worry about it, basically. Okay. Of course, not everybody's gonna have the resources to do that kind of thing, but you know, it is an option for certain people. You can just Google this and learn more about it. I love to tell you more about it. I plan to tell you more about it, but then we don't have time. we have to move on. Okay, so cancer, as I said, is uncontrolled cell division, right? And when you have uncontrolled cell division, in the earlier stage, right, when you just have the oncogene, or if you have a mutated tumor suppressor gene, then the cell is gonna control, divide and become a tumor. Fancy name for the tumor is called a neoplasm, a new growth. Right? That's what a tumor is. Wasn't supposed to be there, but now you have this new growth there, a new tumor. Now, tumor is not necessarily cancerous. Okay? A tumor, if a person has a tumor, you might go and do a biopsy. Okay? A biopsy is, oops, I spelled that wrong. Biopsy, P-S-Y. A biopsy is basically like they take a little bit of a sample of the tumor and then they test it. Right? And the thing that we all want is a benign tumor. Okay? Benign means it's not dangerous. You remove it, goes away, no problem. Okay? So usually that's good news. The bad one is the malignant. Okay? If a tumor is malignant, that means it's cancerous. It's going to progressively get worse. And many forms of cancers, we don't have treatments for them. Okay. But a lot of times, if you discover this early and you do intervention early, then the prognosis improves, right? So how do people know?
Before you go,
Yeah.
So when you do the surgery for the benign ones, does that mean they remove all the cells that are divided?
Yeah, they basically remove the tumor, right? The growth is the cells that have been it's the result of the uncontrolled cell division, right? Yeah.
No, what I'm saying is once the tumor means that the cell is dividing and it's taking over the good cells, right?
The cell is taken over the good cell. Did you say?
Yeah.
Yeah. I mean, they don't really. If it's benign, they don't really take over. Like the good cells are still there, right?
Oh, okay.
So like if you remove the bad ones, then
Okay.
they're gone, right?
Oh,
Yeah.
okay.
Yeah. But if it's malignant, as you will see, it will start to invade nearby tissues, and that's when it's really like you can't really remove all of it, basically. Okay. So after they take the biopsy, right? Like how do they know whether it's benign or not benign? Right? Like what kind of parameters do they use, right? Well, there are 6 characteristics associated with cancer, okay? And they're all listed here. So I'm gonna go through each one of them with you. First 2. Okay. Number 1. Excuse me. Self-sufficiency in growth signal. It means they no longer require external signals to tell them to divide. Okay? You don't need to tell them to divide. They will divide on their own. That's basically what it means. Well, how do they do that, you ask? They have their mutated oncogene, right? They have a green light that's always on. Right? So they got their own signal. They no longer require external signal. Okay, so that's the first characteristics. Self-sufficiency in growth signal. Okay. Number 2, insensitivity to anti-growth signals. They no longer respond to inhibition signal. You tell them to stop, They don't care. They just keep going. Like my kids, right? I tell them, stop hitting each other. Stop fighting. They don't care. They just keep going. They ignore the stop signal. Okay? So number one, they have their own growth signal. And number two, they ignore stop signals. Right? So together, these two things sounds like bad combinations, right? It's already enough to make a cell keep on dividing. Next, they have ability to evade apoptosis. They don't die. Okay? They essentially become immortal. Right? They are able to remember the apoptosis. One way to trigger it is through the checkpoint, right? I was telling you about the checkpoint earlier, the G1 and the G2, right? If you have DNA damage and you cannot repair it, it's supposed to trigger apoptosis, right? But these cancer cells, they find a way to evade apoptosis. So even if you tell them to die, not gonna die. They're just gonna keep on dividing. Now, with these divisions, it requires a lot of energy, a lot of nutrients, right? So when the tumor gets bigger and bigger, another characteristics, another hallmark of cancer is they're able to create their own blood vessel network. Okay? So angiogenesis is creation. It creates new blood vessel. Right? So it basically reroutes the blood vessels and it's sucking up all these nutrients from the surrounding tissue, right? And basically, you know, cancer is taking up all the resources of the body, and that's how it kills the host eventually, right? So has its own growth signal, will not stop growing, even if you tell them to. They can cheat death. They could have new ways to get more nutrients by creating new blood vessels. Last two, they have limitless replicative potential. They no longer become senescent. Why? Because they have telomerase. Okay, the telomerase allows them to reset the telomeres anytime they need. Okay? So the telomeres do not become critically short, and they're able to divide and divide forever, essentially. Okay, and you know this is true. Of course, like in a person, when the person dies, the cells also die. But if you remove the tumor cells, you can grow them indefinitely in a dish. You cannot do that with regular cells. Regular cells, after several division, even if you give them nutrients and stuff, they won't divide anymore. But with cancer cells, they can. And finally, they can invade tissues. Right? They invade the local tissue, and when they run out of space to invade, let's say the cancer starts in the lungs, right? When it's run out of space to invade, then they spread, they metastasize to other organs. Okay, they find a new site and develop. So it's really quite scary, right? And depending on how progressed the disease is, right, how much of tissue has it been invaded, then there is like the stage 1, stage 2, so on and so forth, right? The higher the stage, the more severe the disease is, and the less likely the person is going to survive, right? So those are the 6 hallmarks of cancer. Okay, let's go through these things quickly, and that is it for today. What is a proto-oncogene? Okay, so is it a gene that involves in promoting normal cell growth? Yes, it is. That's what it is. A gene involving in slowing down cell cycle, that is the tumor suppressor gene. A gene that causes cancer, that would be like an oncogene. And proto-oncogene is a gain of function. Right? Remember, these are green lights. It's only going to be problematic if the green lights are always on. Okay, penetrance. You might remember penetrance is the percentage of individuals expressing a gene that they carry. You look at it, 10,000 people in the population. If they all carry this particular gene, how many of them actually express the gene? That percentage would be calculated as expressed as penetrance. The lower the penetrance means the less likely the gene will be expressed. Which of the following is not a tumor suppressor gene? Okay, so a tumor suppressor gene has to do with fixing damages, slowing down growth. So let's see. Responsible for DNA damage, that's going to be tumor suppressor. Telling a cell to die, triggering apoptosis, that's a tumor suppressor. Speeding up the cell cycle, that would not be a tumor suppressor. That would be a proto-oncogene. Okay, checking errors, also tumor suppressor. And that's it. This was a very long lecture, guys. We've got like 72 slides. I understand it's a lot of stuff. Now, for next class, lecture 7, we'll do lecture 7. Lecture 7 is also quite long. I don't think we can finish it next class. So what we're gonna do is we will move the quiz. Let's move it to Wednesday first and see. Okay, I can move the microscope lab as well, so that it's not due on the same day as the quiz. But Wednesday we're gonna have class. I need you to also come next Monday, where we'll do more lecture 7, and I'll also explain the lab to you. And we probably need to have another class on Wednesday as well. Okay, so I will see where we end off on Wednesday first. Definitely we'll move the quiz. I just don't know when to move it. Do you guys have class on Friday, by the way? Like, my understanding is
Yes.
Yeah.
oh, yeah, you do? Okay, is it like a whole day thing or what?
Mine is on site, so.
Oh, okay.
Yeah, mine is on site as well.
All right. Yeah. Okay. So I'll see. Okay. If we end up not finishing lecture 7 by Monday or Wednesday, like maybe I'll move it to maybe I'll open it on the weekend or something. Okay. So that you can have Saturday or Sunday to do it. I know that's not like nice to put things on the weekend. We will see, okay? Maybe I'll put it on Friday all the way to the weekend, so you can choose to do it on Friday if you want to. But I think it's gonna be too rushed if I keep it on Wednesday. Okay, even if we do it on Wednesday, I don't want to rush it. Okay, because 7 is very complicated. And for those of you who don't print your study guide, you need to print that. You need to print your notes, okay, for lecture 7, study guide and the notes, or at least have the PowerPoint so that you can at least print out the PowerPoint so that you can write on it, because it's like math, okay? Lecture 7, a lot of it is like math, where we have to write things down and solve problems. Yeah. Okay, that's it for now. I will see you all on Wednesday. Take care, guys. Bye-bye.
Thank you.
Thank you.